So I'm changing the order around in our schedule just a little bit so that this week we're not directly talking about DNA and identity and then next week human variation. We're going to do DNA and human variation this week, and then we're going to talk about DNA and identity next week. So just so you know, I'm just kind of shaking it up a little bit. So this week we're going to talk about inheritance and evolutionary mechanisms. So for some of you, this is going to be straight out review, and for some of you, this is going to be something you haven't seen in a very long time, and for others, you're just going to have to bear with me to see where we're going, because there is a point. So first of all, let's remember that our DNA is organized by chromosomes, and a chromosome is a nice tightly coiled package of DNA, which when you uncoil it is actually extremely long because each chromosome is billions of base pairs long. The types of chromosomes that we have are two. We have sex chromosomes and we have autosomes. And theoretically, every human has pairs of sex chromosomes and pairs of autosomes, such that your entire genome is made up of sets of pairs of chromosomes. So here is a map of chromosomes, and you can see that with humans, we have 22 pairs of autosomes and 1 pair of sex chromosomes. However, there can be variation in any of these chromosomes, and variation in numbers can lead to a variety of outcomes. But generally speaking, humans have 46 chromosomes, and one of the things that early genome projects showed us is something unexpected, because scientists consider that humans are sort of the epitome of everything and super special, and so we have the most chromosomes because we have the most complex DNA and the most complex genes, etc., etc. However, both chickens and dogs have 78 chromosomes, chimpanzees have 48 chromosomes, and onions even have 16. So the number of chromosomes doesn't actually reflect anything in terms of complexity. It's simply how your DNA is organized. So the pairs that we talked about are called homologous chromosomes. So these pairs, so say your first pair or your 22nd pair, they are the same size and shape, and they code for the same genes. Alleles are versions of genes on homologous chromosomes. So you get one set of chromosomes from your mom, the other set of chromosomes from your dad, and at pair 22, for example, is the information that codes for hair color. That's a silly, dumb example, but let's go with it. And so there's the gene for hair color at chromosome 22, and you get one allele or version from your mom and one allele or version from your dad. And that's how it works for all of the genes in your body. Now the genotype is your genetic makeup. So basically this is how we refer to the alleles of an individual at a particular locus, which is the location on a chromosome, so the address on a chromosome. And as an example of genotypes, we use big letters and little letters. So you may be big A, big A. You may be big A, little a. You may be little a, little a. Those are the combinations of alleles that make up your genotype. A phenotype is the observable or measurable feature of an organism. So it can be anatomical, biochemical, behavioral. Basically, the genotype is what DNA you have, what alleles you have. Your phenotype is what you look like, or you sound like, or how you act as a result of that genotype. Something else to remember is that not all genotypes code or result or are represented by phenotypes in the same way. And so when we are looking at the potential combinations of alleles, if we're using big A's and little a's, the reason we are doing that is because some alleles are dominant over others. So we represent dominant and recessive by the big A and the little a. So dominant is big A, little a is recessive. So little a is something called homozygous recessive. So homozygous means two of the same, and so homozygous recessive means two little a's, essentially. Or you could use B's or C's, whatever letter you're using, two little versions of that. And in our example here, we're going to follow a famous pea plant example by Gregor Mendel. This phenotype is green for our peas. Now, if we go down here, we can think about big A, which is homozygous dominant. So again, that's two of the same. So homozygous means two of the same. And dominant means the big allele, or the dominant allele. So homozygous dominant and big A little a is having two different ones. So the word we use for that is heterozygous. So this genotype is heterozygous, which means it has a dominant one and a recessive one, a big one and a little one. For both of these genotypes,
Something happened to its screen. Like it went to the screen, and it
sorry about that. My daughter and I have a walkie-talkie now that every once in a while she talks on while she's in school, so I apologize for that, but I've already recorded so much. I'll try and cut this out if I can. What I was saying is that both for homozygous dominant and heterozygous, the phenotype is yellow because anytime a dominant allele is present, it is expressed. So in our example here, the dominant allele is yellow, expresses as yellow. The recessive allele expresses as green. But the only way you get the green phenotype is if you're homozygous recessive. And the only way you get the yellow phenotype, is if you're homozygous dominant or heterozygous, because either way you have one of the big alleles. You have one of the dominant alleles. Now, a cool part of this story is that Gregor Mendel was a monk who actually had figured out all of that stuff about genotypes and phenotypes and alleles long before we knew what the structure of DNA actually is. So we're going to go into very few details about how DNA is structured, because we're all leading to a certain spot. We're all getting to the point where we can understand more about variation in modern humans. So DNA is short for deoxyribonucleic acid, which is basically ladder-shaped. Sometimes I think of it as zipper-shaped, but a ladder with two sides of the ladder and rungs across the middle. And Watson and Crick were two scientists who were given the Nobel Peace Prize for illustrating the structure of DNA through X-rays and photos of X-rays. However, it was Rosalind Franklin who, with her ability to take X-ray photographs, was the one who actually saw the structure of DNA that they then described. And so it was this famous photo 51 here that she took that was really why they were able to do it. And she, of course, died before they got the Nobel Prize. She died of radiation poisoning from doing all the X-rays, so she was never given that Nobel Peace Prize herself. But the very kind of simple details about the structure of DNA, if we think about it as a ladder, a twisting ladder, what we can say is that there are basic building blocks of that ladder. And the building blocks include part of the sides of the ladder as well as half of a rung, because each side is going to be part of a puzzle that goes together and meets in the middle of those rungs. So a nucleotide is the basic building block. And each nucleotide includes a sugar molecule and a phosphate molecule that are both part of the structure of the sides of the ladder, so that backbone, the sugar phosphate backbone, and one of four bases. And the bases are guanine, cytosine, thymine, and adenine, and we usually just talk about them as G's, C's, T's, and A's. So if you look down here, what we have is the phosphate and sugar backbone, and then this C here is a base, and all three together make up the nucleotide, and DNA is just a series of these nucleotides stacked on top of each other. They also meet up in the middle with another series of nucleotides stacked up on top of each other. Now, when you look at this particular representation of this ladder, this is untwisted, what we can see is that each nucleotide here with the phosphate and sugar backbone and the base in the middle has to match up with a specific partner. So those partners in the center of that rung are called base pairs. And they always partner in this way. C's and G's partner together and A's and T's partner together, because that's the way that the bonds will work. So no other way will these partner. So this is what keeps the basic structure of DNA together. And the reason we're talking about the structure of DNA is because the jobs that DNA has in our body is incredibly important for how our body functions, but also for understanding variation in our genome and variation in our population. So the primary functions of DNA are simply to replicate. So this is the process by which DNA is copied. It has to be replicated during cell division. And so this is just a simple way of making sure you have the same version of DNA in every cell. Transcription is the first step in how proteins are synthesized. So this is the process by which information in DNA is transferred to messenger RNA, or mRNA. And we're going to go into some details about this just to remind you. And translation is the second step in protein synthesis. So this is the process by which information in that messenger RNA, that mRNA, is utilized to create proteins. So basically, the two primary functions of DNA are simply to replicate, and then they are carrying the information that's needed to create proteins in your body, to make proteins in your body. And that's where we're going to get into some interesting stuff for humans. So as I mentioned on that slide, DNA replication is fairly simple. It's as if the two sides of your ladder unzip, or that's the zipper, but they come apart, and then new strands are formed. Sorry, so what I was saying is that the sides of the DNA unzip and new halves of DNA come in. New halves of DNA formed by new nucleotides come in to match up, to partner up, pair up with the bases that are already there. So basically you get two exact pairs of DNA where you had one before. And this happens during cell replication. Now the meat of the story, though, is going to be in talking about proteins and protein synthesis. So just as a little refresher, proteins are found all over your body. Your hemoglobin is for oxygen transport. Your collagen is your connective tissue, for example. Actin and myosin are muscles. I know that proteins and hormones have a whole other thing around them, but things like adrenaline is good for metabolism. And your body, the cells in your body, synthesize all of these proteins. So they produce all of these proteins. And the DNA in every cell has all the information that's needed to produce these proteins. The basic building blocks of proteins are amino acids. So 20 amino acids that your body needs to build these proteins, to make these amino acid chains that are then functioning proteins. 9 of them are essential amino acids because you actually have to consume them. Your body doesn't produce them. So it's important to make sure you have those in your diet so that your body has all of the building blocks it needs to synthesize functional proteins. And a primary amino acid sequence, we can visualize it kind of like this, kind of like a string of beads or a train with cars connected together, and each one of those is one of those amino acids. And eventually that primary amino acid sequence will fold to form some sort of functional protein. So this example here, it actually has multiple amino acid sequences folded together to make the eventual functional protein. So information is often needed from various different genes in order to create all of the strands that are necessary for a functional protein. Now, one of my favorite things about this is that different cells can make different kinds of proteins. They do make different kinds of proteins. So cells in your eyes are going to make the protein rhodopsin for light detection, and cells in your pituitary gland are going to make the proteins that are necessary for growth hormone. So even though every cell in your body has the full complement of DNA, except for your sex cells, those are a little bit different, has the full complement of your DNA, your 23 pairs of DNA, or 23 pairs of chromosomes, the cells in various parts of your body only make the proteins that are needed for functioning in that part of your body. So as I mentioned, protein synthesis has two primary steps, transcription and translation. And then after those two steps, you have the formation of a functional protein. So that little circle I have here is supposed to be a cell, and then the nucleus is there in the center in darker orange. So transcription takes place inside the nucleus, translation takes place outside the nucleus. This happens the same whether it's a liver cell, a nerve cell, or a muscle cell. The 46 chromosomes are all there, and transcription, translation, and general protein synthesis can happen there. They just happen for the proteins that are needed in those areas. So one of the fun questions is, how do we get from a sequence of DNA, which is where we started, to a sequence of 20 different amino acids, or to our protein amino acid chain? So up here at the top of the slide, we have a representation of 1/2 of a DNA molecule. So this up here, these are the sugar and phosphate backbones, and then these are the bases. So this isn't both halves of the ladder, this is 1/2 of the ladder. So how do we go from that information to this sequence of amino acids. It's cool and fun. When we look at these sequences of base pairs, what we can see, or bases, what we can see is that 3 in a row represent something called a codon. And a codon codes for a specific amino acid. So in this case, CAA is coding for GLU. And when we look at all the sequence of bases, we can see codon after codon after codon. Each codon codes for a particular amino acid. So it's kind of like just using a decoder ring to say, these are my three bases, 1, 2, 3, and that codes for this amino acid. Here's my next 3, 1, 2, 3 bases, and that codes for this amino acid. And that is sort of sillily called the genetic code, how the codons code for the specific amino acids. So here is a fancy slide with lots of information that you will never need again, likely. But these are 3 bases in a row, and there's one base pair difference in this because this is talking about a specific type of messenger RNA. But you can see that each one of these codes for a particular amino acid and that there is redundancy in the system. So it's important that you have more than one set of bases that code for a specific amino acid. This cuts down on errors. However, it doesn't remove errors, which is where we're going with this. So as I mentioned earlier, transcription happens within the nucleus and translation happens outside of the nucleus. Because translation happens outside the nucleus, the DNA needs help because the DNA cannot leave the nucleus. So this is where the messenger RNA comes in that we mentioned earlier, that DNA inside the nucleus is read by messenger RNA. It takes the message outside of the nucleus where it can be translated into an actual polypeptide, which is an amino acid chain. So transcription happening inside the nucleus is when just a particular part of the DNA is unwound. This is basically where the gene is. So the DNA pulls apart at this area, and then other nucleotides come in to form a corresponding sequence. So remember, they have to pair up in a certain order, and this corresponding sequence, this mRNA, is the opposite, so the exact opposite, the pairs that are necessary to form corresponding to the information on the DNA. So mRNA is the messenger RNA that is read from the DNA in the nucleus during transcription. There are some big differences between DNA and RNA. DNA, you can see on the left, is double-stranded. It's deoxyribose sugar and thymine, so deoxy means 2. And here on the right, you see RNA, which is single-stranded, and so it has 1 ribose sugar, and it also specifically has uracil. So I might have said thymine a second ago. But thymine is the base that you have here on DNA, and on RNA there is no thymine, there is a uracil instead. So anytime you see a sequence of bases that includes a U, you know it's RNA and not DNA. And this is the only one that's formed for DNA. It's single-stranded, and that way it can move outside of the nucleus and take the information with it. But if you see here, this is the part of the DNA that has been read. This is what the normal second half of the DNA looks like, but this instead is the RNA that's read that information, and so C as opposed to G, U as opposed to A as opposed to T, and G as opposed to C. So they still pair in the same ways. That mRNA takes that message outside of the nucleus to be translated. But first it's edited. So if we think about DNA in the nucleus, there's a whole lot of DNA, and not all of it has the information we need. So first it's read, it's transcribed, and then the mRNA is basically chunked up into what are called exons and introns. Exons are the information we actually want to be translated. Introns are not. It's extra information that's not used in the translation process. So the exons are edited together into your mRNA strand that then leaves the nucleus to go be translated for protein synthesis. So what we have here is a strand that's going through the ribosome here and being translated. Here is another way to look at this, and a reminder that an exon is a section of genes that codes for a portion of an amino acid chain of a protein. An intron is a section of gene that does not code for a portion of a protein. So you can imagine, if we're going to talk about errors after a bit, which is where we're going, that it could theoretically be not too hard for errors to happen when identifying exons and introns. So your ribosomes, and I'm sure you didn't expect to see this figure outside of 5th grade again, your ribosomes are your little powerhouses that are creating proteins that are all over endoplasmic reticulum that surrounds your nucleus where your DNA is hanging out. So the messenger RNA can leave the nucleus, brings the information to one of these ribosomes where a protein is synthesized. Now it actually needs help to synthesize that protein. So mRNA helps the DNA in the nucleus so that it can leave the nucleus, and then tRNA is called transfer RNA, and it is what brings the amino acid when it's supposed to come. So we represent them so they look a little bit like a T here, and what it is again, this is a string of bases, but there's U's because it's RNA and it's single-stranded, and it's organized in such a way that it has a acceptor arm that holds on to an amino acid. So up here at the tail, it carries around an amino acid. And down here at the head, there's something called an anticodon. So each codon codes for an amino acid. The tRNA has the anticodon, has the opposite set of pairs so that it can match together, they can pair together, and it has this little amino acid tail that it has following behind it. So it floats around with an anticodon looking for a place to dock, bringing its amino acid tail with it. So what happens during translation is that the ribosome goes along the messenger RNA here. I kind of think of it as a train going through a tunnel kind of thing. And so the train goes through the tunnel, and when it's inside the tunnel, what happens is that the tRNAs will come down, will dock where it's appropriate, given the codon and the anticodon, and bring this amino acid with it. Then the amino acids connect to each other so that you have a polypeptide amino acid chain. So our examples here, for instance, these are the codons, GGG, GCA, GUU, and these are the anticodons that are the opposite, that are the match, so that the tRNA puts the polypeptide in the right spot. So all of this information is available on the DNA. It's first read by the mRNA, which brings the message, and then in the ribosome, the tRNA, the transfer RNA, brings each amino acid in the correct order to create that primary amino acid sequence that eventually folds to be a functional protein. So this is just the reminder that transcription happens inside the nucleus. The mRNA takes the message outside of the nucleus to the ribosome, where translation happens and tRNA brings the peptides, the amino acids, to help then form that polypeptide chain. Also, as a reminder, you start out with the primary structure, which is the polypeptide the polypeptide chain, the single strand of amino acids that then fold together in some sort of tertiary structure to be that functional protein. This happens in all of your cells, and cell differentiation is basically cells taking up the jobs that they will have. So not that long after fertilization, when the zygote is formed and the cells are rapidly dividing, they take up different jobs as the embryo develops. So looking at human development, stage 13, human embryo, about 28 days there on the left, and then about 54 days down towards the bottom, you can see clearly that cells already have various different jobs and are producing the proteins that are needed for that region of the developing embryo. So back to cell division for a moment, when DNA is simply replicating. There's two different types of cell division. Mitosis is somatic cell division. That's your body cells dividing, when one cell divides into two identical daughter cells. So that's where you get the single strand of DNA replicating into two exact same copies of that DNA. So you go from one cell to two cells. The DNA has to go from one version of it to two versions of it. Version's not the right word. Copies. One copy of it to two copies of it. Meiosis is the cell division that's a little bit more interesting. It leads to the formation of gametes, which are your sperm or your ova. So this only happens when you are creating sex cells. Now, a mutation can happen during mitosis or meiosis. A mutation is any error that occurs in the process of copying DNA that can be transmitted to daughter cells. So duplicating DNA can cause errors. Any error, any missed DNA, missed base, anything that can be transmitted to daughter cells, any error is called a mutation. They can occur during both mitosis and meiosis, and I know that you guys who have more recently had this know a million different phases of mitosis and meiosis. We're just not going to worry about that. We're going to worry about the fact that mitosis, when a mutation happens, it goes from one parent cell to two daughter cells. Those are body cells. Those are the cells that basically make up your toes and your hair and everything. Somatic cell mutations aren't inherited. You don't pass those down to your offspring. During meiosis, though, you have one cell that ultimately forms your gametes, either your sperm or your ova, and those will be passed down to your offspring. First, though, here's somatic mutations at cancer-causing loci. So this is an old figure now, but each one of these red arrows indicates where a mutation in just a regular genome could cause cancer. These are errors that happen in an individual's body in their somatic cells during mitosis. These are not errors that are going to be passed down to their offspring. So that's how people develop non-hereditary cancer. Where I got ahead of myself a little bit was talking about meiosis and the developing of gametes. If mutations occur during this cell division process, then mutations are in your gametes and are passed down to the offspring. Now, mutations are rare. So there's only this many mutations per nucleotide per generation. So you think, oh my goodness, that's like nothing. But there are 3.2 billion nucleotides in your genome. So on average, each of us has mutations in about 58 nucleotides compared to our parents. So you have about 58 errors in your DNA compared to what your parents had before they basically gave you your DNA. So 58 sounds kind of like a big number. However, if you look at the genome here, and so this, we're talking about this long line here, this is the non-transcribed region of your genome. This is the transcribed, so this is this part that's actually read and kept in your mRNA. And then this red bit at the end is the part that's actually transcribed and translated. So that's the part that's actually used for synthesizing proteins. So for 58 nucleotide changes, they'll all be passed on to the offspring, but the only ones that are actually going to do anything to you that you would know about or would change the function of anything in your body are the ones that are actually in the transcribed and translated regions of the individuals who are carrying them. So for the vast majority of us, those 58 nucleotide changes are in that non-transcribed region, and we never know. There's no reason to know. Certain mutations, however, can have pretty big effects. Even point mutations, which is one change in a base. So if we're looking at this sequence up here, this is the normal beta hemoglobin sequence. This is the sequence of bases that makes a set of codons that codes for a chain of polypeptides or a chain of peptides, chain of amino acids that together make a functional protein. Normally, in normal beta hemoglobin sequence, you have a T here, but in our example of a point mutation, that T has been replaced by an A. So that's one error in one spot on the whole gene. And what happens is that one error changes the codon, which then changes the amino acid that is coded for. Now, there is redundancy in the system, so often, the majority of the time, point mutations don't have any influence on the peptides that they are coding for. But in this case, that point mutation, that one base mutation, does code for a different amino acid, which in this case, instead of a normal beta hemoglobin sequence, becomes a sickle cell beta hemoglobin sequence. And the person who has this one point mutation has sickle cell anemia. So those who have sickle cell anemia, that means their blood cells form this sickle shape, and that can be very painful as they kind of build up in organs, and it can also mean that oxygen does not move throughout their blood system as easily. So this is something to be aware of. And what's amazing, though, is that it's the result of one point mutation. Kind of thinking about it the opposite way, single cells usually do influence several traits. So a gene at this particular locus might influence trait A, B, and C. This is called pleiotropy when one gene has multiple effects. So if you imagine if there's one mutation on that gene, it's not just going to influence one thing. It's going to influence all of these things. It's going to influence the synthesis of multiple different proteins. Most proteins are also derived from multiple polypeptides, and thus multiple genes. So there are more than one chance for a mutation to influence a protein. So it's not necessarily that you have a mutation on chromosome 11, you might have a mutation on chromosome 16, and that will influence how that polypeptide is formed, and thus influence the protein overall. Now, when we talk about traits and how traits are passed down, we can kind of put all of this information together and think about human variation. And some traits that are variable in humans are called simple Mendelian traits, and some traits that are variable in humans are called complex traits. The simple Mendelian traits are located in one place on a chromosome, so there's only one gene coding for them, so they're called single locus. They only have two alleles, and they have a simple dominant recessive system. Complex traits are the ones that we much more often see and talk about. They have multiple loci, so they're polygenic. There's multiple genes responsible for information that's coding for these polypeptide chains that create these functional proteins. So you find the information you need at multiple places on chromosomes. Many alleles are involved in the expression. There can be something called partial dominance. So at the very beginning, I was talking about heterozygous and homozygous dominant, homozygous negative, recessive, as if that was the only way that alleles interact with each other, and it's not. Partial dominance means that maybe both alleles are expressed. So if you have a yellow allele and a green allele, yellow and blue make green, yellow allele and a blue allele, if there's partial dominance, you might see green rather than either yellow or blue. And for humans, we have a couple examples of simple Mendelian traits. These ones are only found at one gene, only two alleles, simple, dominance, and recessive. Things like having a widow's peak is Mendelian, you either do or you don't. Having attached earlobes is Mendelian, you either do or you don't. And I have the most attached earlobes on the planet, so I'm sorry that I can't show you those. And so the distribution of these is called discontinuous. So this is qualitative variation. You either do or you don't. You don't measure a number or a metric. It's either yes or no. And so qualitative variation is always discontinuously distributed. It means there's a group of people in the yes column and a group of people in the no column. That's discontinuous. And it depends on whether you have a dominant or a recessive allele. Complex traits are continuously distributed, which means they are quantitative in their variation and basically can go from essentially little bitty to giant and everything in between. So my example here is bird beak depth, and so there's going to be a few birds with teeny tiny beaks and a few birds with big giant beaks, and there's going to be a whole bunch of birds with sort of middle-sized beaks. But it's continuously distributed. It means it has this bell curve because there's representatives from every size in between the largest and the smallest. Most human variation that we talk about is quantitative variation that's continuously distributed like this. Height in humans is a good example. We have an average female height, an average male height, and then together we have an average human height. And you can see that basically there's the teeny tiniest and the biggest, and then everything in between is also represented. So continuously distributed. Now for continuous variation, this quantitative variation, if we have the phenotypes here, the genotypes that are being expressed are going to be on multiple loci, so basically multiple locations on different chromosomes, so that also means multiple genes, and multiple alleles that are coding for those genes, so multiple different versions, multiple different alleles. So here you have A's and B's and C's and D's, and all of these together are going to be responsible for coding for this complex trait. So it's no longer just A, big A, little a. It's now A's and B's and C's and D's and etc. And some genes control other genes. So structural genes code for the proteins. Those are the ones we've been talking about this whole time. And mutations in structural genes are going to result in mutations potentially that influence the synthesis of the proteins. But potentially even more important or more is regulatory genes. And regulatory genes influence the expression of these structural genes. So basically, a regulatory gene is not telling a protein how to be synthesized. It's telling another gene when to synthesize, or how much to make, or where to make it. So a famous example here is Antennapedia and Drosophila. These are fruit fly faces. And what scientists have done is normally where there would be antenna, they got a regulatory gene to basically turn on the message that said, grow feet. So feet here are coming out of where the antenna would normally be, not because of a mutation in the structural gene, but because of a mutation in the regulatory gene. So basically a mutation that told a different protein when to turn on, when to turn off, how much to make, where to grow, where to build. And so mutations can occur that will influence both structural genes and regulatory genes, and both of them may have long-term effects. Mutations in either structural genes or regulatory genes are going to lead to something important. They're going to lead to variation in the population. So unless DNA essentially makes mistakes, or mistakes are made in the transcription or the translation or the replication of DNA, then there aren't going to be any changes in the system, and there's not going to be new variation. So new variation comes from mutation. Mutation is critical for the system. And people like Darwin and others noticed this variation in the population and how important it is, and that's what led Darwin to saying individuals with features that allow them to outcompete others for limited resources will be more fit, which means they will reproduce more and have more successful offspring, and that over time the population will change to resemble the most fit individuals. So variation, and the variation that comes from mutation, that's why we've gone through this whole thing today, is so that we can see that mutation is necessary for variation and variation is necessary for natural selection. That's the basis for the evolution or the change of species over time. So our definition of evolution is a change in allele frequency over time. It's a change in the frequency of alleles between generations. And alleles, these versions of genes where we get new alleles from, is mutation.